Long before eyes could see a sunrise, life faced a problem. Light returned with extraordinary regularity. A cell could wait for the first rays and react, or it could prepare for morning before morning arrived. That difference sounds small. It is the difference between a light switch and a clock. A switch responds to what is happening now. A clock carries information about what is likely to happen next. The setting was an Earth almost beyond recognition. Oxygen-producing photosynthesis evolved in ancient microbes, including the ancestors of cyanobacteria. But the familiar date of about 2.4 billion years marks a major rise of oxygen in the atmosphere, not a precise birthday for photosynthesis.
For a long time, oxygen released into water met iron, volcanic gases, and other chemical sinks. Only after many such sinks were overwhelmed could oxygen accumulate more widely. The transformation was vast, but it was not a single afternoon when the planet suddenly turned blue.
For a photosynthetic cell, day and night divide the work. Light can power carbon fixation. Darkness changes the energy available, and in some cyanobacteria, helps separate oxygen-sensitive nitrogen fixation from oxygen-producing photosynthesis. Timing can prevent incompatible chemistry from colliding.
Scientists do not call every daily response a circadian clock. A genuine clock must pass 3 tests. It can be reset by cues such as light or temperature. It keeps running in constant conditions, and its period remains fairly stable across ordinary temperature changes.
Cyanobacteria pass those tests. Their timekeeper is especially revealing because its central machinery can be taken out of a living cell and studied piece by piece.
At the center is KaiABC, a protein assembled as 6 matching units in 2 stacked rings. It uses ATP slowly. During one cycle, 2 sites on each unit gain phosphate groups in a fixed order, then lose them in the reverse sequence. KaiA favors the phosphorylation phase. Later, KaiB changes shape, binds KaiC, and helps trap KaiA in a nighttime complex. KaiC then moves through dephosphorylation. This is not a tiny clock face. It is a population of proteins changing state together.
In 2005, researchers performed an experiment that still feels improbable. They purified KaiA, KaiB, and KaiC, mixed them with ATP in a test tube, and kept the conditions constant.
KaiC phosphorylation rose and fell with a period close to 24 hours. The rhythm continued without a genome being read and without new proteins being made. It was also temperature compensated. Much of the clock's core timing was chemistry.
The tube did not contain a complete living clock. Cells add sensors, gene control, metabolism, and repair. But the experiment isolated something fundamental. 3 proteins and an energy source could generate a persistent daily rhythm.
A clock is elegant. Does it help? In a 1998 competition, scientists mixed cyanobacterial strains with different internal periods. Under a 24-hour light-dark cycle, the strain whose clock best matched that external rhythm tended to win.
Change the length of the artificial day, and the advantage changed with it. The best clock was not simply the fastest or slowest. It was the one in resonance with the environment. This is what it means to say that life learned the rhythm of dawn. No cell understood tomorrow. Across generations, variants that scheduled their chemistry more effectively left more descendants. Anticipation accumulated without foresight.
Even so, the modern KaiA, KaiB, and KaiC system should not be projected unchanged into the deepest past. In 2025, researchers reconstructed ancestral Kai proteins from evolutionary family trees. Their results suggest that useful timekeeping appeared in stages. One reconstructed system, assigned by the authors to roughly 950 million years ago, could be reset by environmental cycles, but did not sustain a free-running rhythm in the tested tube conditions. In engineered cells, it performed better under the shorter day, the researchers associated with that era.
Those dates are model-dependent, and reconstructed proteins are experiments about plausible ancestors, not fossils brought back to life. Still, they challenge a tidy origin story. A resettable timer may have come before a self-sustained clock.
Not every cyanobacterium carries the full KaiA, KaiB, and KaiC arrangement, and the clocks of plants, fungi, insects, and mammals use different molecular machinery. Daily timekeeping is widespread, but evolution has found more than one way to build it.
Inside one cell, every molecular collision contains noise. Yet millions of slow reactions can synchronize into a reliable phase. Morning, evening, night, and morning again. Order does not require each molecule to be precise. It can emerge from how the molecules correct and constrain one another. Life did not invent the day, and it did not know that sunrise was coming. Selection built chemistry that could carry a memory of recurring light into darkness. Before life could see the dawn, part of life had already learned how to be ready for it.